In this video we will discuss about role of SLIT2, ATF4 and CGRP in tumor suppression. So in the previous video we have understood how this tumor suppression will be dependent on the different population of immune cells in the tumor derived lymph node, and the major populations that got changed in the presence of chemogenetic inhibition is the CD8 plus T cells, gamma delta T cells, and activated conventional dendritic cells. So their focus is on these three populations mainly to find out the role of SLIT2, ATF4 and CGRP in tumor suppression. So first they started with this SLIT2. So what it did is they make the SLIT2 knocked out and then check the effect of CD8 plus T cells in the presence of this chemogenetic inhibition agent HM4 DGI. So in the control they are able to see an increase in the CD8 plus T cells, and also when they inject intratumorally inject recombinant SLIT2, they are able to see further enhance in the difference between the control and the this intratumoral injection of recombinant SLIT2 is high. However, when the SLIT2 knockout is there, the difference is not significant. And when they in the SLIT2 knockout, when they intratumorally inject recombinant SLIT2, the difference is statistically significant. And when they did the same analysis for the gamma delta T cells and activated conventional dendritic cells, they are able to see similar results. That is, in the presence of SLIT2 knockout, there is no difference for gamma delta T cells and activated conventional dendritic cells. However, when they intratumorally inject recombinant SLIT2, there is an enhanced signal or increase in the gamma delta T cells and the conventional dendritic C cells, indicating that SLIT2 has a role towards this tumor suppressions. And afterwards they checked it. They did the similar studies on the ATF4. That is, they make the ATF4 knockout and check there is no statistically significant difference between CD8 plus T cells, gamma delta T cells, and activated conventional dendritic cells. However, when they intratumorally inject recombinant SLIT2, there is a difference in the CD8 plus T cells, gamma delta T cells, and conventional dendritic cells can be seen. And with this, immune cells population are high in the presence of chemogenetic inhibition using HM4 DGI. So once validating the role of SLIT2 and AT4, now they also check the results for the CGRP. The CGRP protein is produced by this gene calca. So they knocked down the calca and then checked its effect. In the presence of calca, they are able to see increase in the CD8 plus T cells and also increase in the gamma delta T cells and the conventional dendritic cells. So because CGRP is also from the previous results seems like plays a role towards tumor inhibitions. So in the absence of this CGRP, these immune cells also goes up, and the other immune cells also goes up, that's shown here. This NK cells, macrophages, monocyte, neutrophil, eosinophils, and plasmablast there is no change. Afterwards, they did it for panel. They did it for the case where they inject this CGRP, and when they inject this CGRP in the chemogenetic inhibition method, and they use both the method, that is HM4 DGI and RTX based method. So when there is no CGRP, only saline is injected, they are able to see more CD8 plus T cells are there, and more gamma delta T cells and activated conventional dendritic cells are there, compared to the case where they inject CGRP, intratumorally inject CGRP. You can see that CD8 plus T cell signal goes down for both HM4 DGI and RTX based method, and similar results can be seen for gamma delta T cells and activated conventional dendritic cells for both HM4 DGI method and RTX based chemical chemogenetic inhibition method. So this in all together indicates that in the absence of either of this SLIT2, ATF4 or CGRP, the tumor suppression process can be inhibited, and these things they are validated individually. And if this SLIT2, ATF4 and CGRP are intratumorally injected, then we can see that there is a rescue in the tumor suppression operations through the decrease in the immune cells that are decrease in the activated immune cells such as CD8 plus T cells, counter T cells, and activated conventional T cells goes down in the tumor derived lymph node. This shows the significant role of all SLIT2, ATF4 and CGRP in the tumor suppressions using innervating using activation of innervating nerve. So that's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.